In this third video of this series about exocrine glands, we will start dealing in detail with each of the classification criteria for these glands. We already know that exocrine glands maintain the connection with the original epithelial lining, thus releasing their products of secretion in the lumen of an organ or on the surface of the body. In this video, we will go deeper in the answer to the first of the 5 questions presented in the previous video, and we will learn how to identify the ducts in the histological section slides. The remaining questions will be addressed in the same way in the next videos. The histological issue addressed in this video is: in a certain exocrine gland, are we able to observe only a single duct, or are we able to find several ducts? If the exocrine gland has only one single undivided duct, it is called simple. Simple straight tubular. Simple coil tubular. Simple acinar. Simple alveolar. Simple branched tubulo-acinar. But if the duct is segmented into others, the exocrine gland is classified as compound. Compound branched tubulo-acinar. Compound branched tubulo-alveolar. We find examples of ducts that do not divide on the esophagus slides. One of them has already been presented in the first video of this series. In this section, we notice that although the existing exocrine glands in the esophagus wall have a well-developed secretory portion, each of the secretory cell clusters will only connect to a single duct. When observing a histological slide, we cannot always fully see the duct that connects the secretory portion of each esophageal gland to the epithelial lining. But we do see many fragments. This happens because the ducts of these glands are not straight structures. They make winding pathways. When going to a salivary gland slide, we find ducts of different sizes. Some of them are located in the septa, as in this septum we see in this image. The septa are invaginations of the connective tissue of the capsule. They delimit the lobules formed by the secretory portion clusters. Other ducts are immersed in these secretory portions. Inside of some ducts, we are able to see the secretion stained in Gaussian blue, used to highlight the acid mucins of the saliva. The main duct penetrates the gland by the hilum and divides into ducts of smaller calibers throughout the septa. The ducts immersed in the septa, like this one, are called interlobular. The epithelia of interlobular ducts may be simple columnar, pseudostratified, or even stratified squamous, due to the thick ductal caliber. When the minor segments penetrate the lobules, we have intralobular ducts, covered by a simple epithelium or by 2 layers of cuboid or columnar cells. The intralobular ducts also continue to segment and decrease in caliber until they communicate with their due secretory portions, from which they collect the secretion produced. The larger exocrine glands, which end up forming whole organs like the pancreas, the liver, and all the large salivary glands, have a similar composition. Hence, this general structure we have presented may be applied to all the other larger exocrine glands. But be careful not to mistake ducts for blood vessels that also run on the septa, but have the endothelium composed by one single layer of squamous epithelial cells. Sometimes it is possible to see the red blood cells in some vessels. We have then seen in this video that the simple exocrine gland is the one whose duct does not divide and is directly connected to the secretory portion, while the compound exocrine gland is the one whose main duct is divided and in which the cells of the secretory portions connect to ductal subdivisions. In order to go deeper in the classification of the exocrine glands according to the secretory portion, watch the next video.